Today I talked about the work that we've been doing in my lab and with my group over the last 10 years to try to understand the immune microenvironment in ovarian cancer and using ovarian as a model to understand solid tumor biology in general. So we have, since 10 years now, found that in ovarian cancer there is a spontaneous infiltration of T cells. So there is a spontaneous adaptive immunity, a sort of a spontaneous immune recognition in an attempt to reject the tumors in a proportion of patients. We have been able to correlate that with a very strong biomarker, which is the presence of intraepithelial or intratumoral T cells. By the way, the same biomarker has been identified in many other tumors, notably in colorectal cancer and many others. And it turns out that the presence of these T cells in the tumor predicts significantly better overall survival following conventional treatments. In fact, in our series, we found that the only patients who survive long-term are the ones who have these T cells at the time of diagnosis. Now, importantly, these T cells, this phenotype is quite robust and persists even after chemotherapy, which explains the long-term benefits. And we recently looked at the whole literature with over 1,800 patients and found in ovarian cancer, over 10 papers, and found that they all basically went in the same direction, showing that they were all concordant with an increased hazard ratio for risk of death if one does not have these T cells. Now, this is very important biologically, but it's also very important therapeutically because it really reveals that there are two kinds of patients when it comes to immunotherapy. One group of patients in which there is already an immune response that tries to reject tumors. And there, what we need to do is understand what prevents it from doing so and basically helping it to do that, basically unleashing those inhibitory barriers. I spoke today about the PD-1, PD-L1 pathway, which is, we think, in ovarian cancer and in many other tumors, a very important pathway, but not the only one for sure. And it's clearly shown in mouse models and in vitro systems with human tumor microenvironment reconstitution in ovarian cancer that when there are T cells in the microenvironment, tumors do respond to PD-L1 blockade or double PD-L1 and CTLA-4 blockade, and that is a very clear pathway for clinical development. And in fact, presently there is a clinical trial that will be conducted in the context of the cancer vaccine cooperative group, which will test exactly double blockade in a cohort of ovarian patients. In addition, the question becomes what happens with patients who do not have T cells at the onset. And there, I think it's critically important to understand what keeps the T cells out of the tumor. And is it because the tumors are not immunogenic, or is it because the T cells cannot be allowed to get in? And we found through mouse models and human studies that the latter is true, and that is that tumors are potentially immunogenic, but T cells are not allowed to home into tumors to establish a rejection response. And the vasculature is partially responsible of that through a system of mechanisms that we collectively call the tumor endothelial barrier. So effective T cells cannot cross that barrier to get into the tumor. Same as it happens with the brain, for example, which becomes an immune-privileged site. So the tumor similarly establishes immune-privileged territory, not in all patients, but in some of the patients where there are no T cells at the time of diagnosis. Certainly this could be one of the reasons. And now we found mechanisms to basically counter this pharmacologically. We identified two molecular pathways, and both of them can be pharmacologically suppressed. And I spoke about our recent data that was published where we found that the tumor endothelium expresses a death ligand called Fas ligand. And that's maintained in tumor vasculature through a cooperative action of VEGF, IL-10, and prostaglandin E2. So by using commercially available bevacizumab, for example, or other VEGF inhibition and prostaglandin inhibitors, such as the old nice aspirin or indomethacin and so forth, you can take that barrier down and allow T cells to get into the tumor. And now for the first time, we found ways to restore T cell infiltration in tumors, which could be an important step towards preparing patients for PD-L1 therapy. And so the opportunity now is that we could potentially use PD-L1 or other types of this T cell stimulating pharmacology to also for patients who did not have T cells in the tumors at the beginning. We found T cells infiltrating in every stage of ovarian cancer, including stage 1, 2, and so forth. We've done extensive studies, of course, in stage 3 and 4 patients because these are the most common variety and the most interesting from the standpoint of identifying new therapies, as they are the most challenging in the clinic to treat. Notably there, we found that in a series, the only patients who are cured, so to speak, after chemotherapy, so they have long-term remissions, are the patients who had these T cells to begin with. Yes, so cervical cancer, being a virally induced tumor with well-integrated viral antigens into the cancer cell genome, which are mandatory because they are part of the oncogenetic process. These oncogenes, the HPV6 and E7, are integrated in the cancer cell genome, and they are always expressed, so the tumor cannot eliminate them. And so these are epitopes that the immune system can recognize, and there are in fact responses against these, and there are tumor-infiltrating lymphocytes in cervical cancer. So I think cervical cancer is a very good opportunity for immunotherapy, and in fact underexploited at the moment. But we're working with different companies to develop PD-1 based therapeutic approaches for cervical cancer and vaccines as well. Uterine cancer is also very interesting. A proportion of uterine cancers are linked to the Lynch syndrome, and so they have microsatellite instability. So it turns out that those patients, those tumors, have hundreds of non-synonymous mutations. It's much like colorectal cancer linked to the Lynch syndrome. And so those patients, those tumors, actually do have a very florid T cell infiltration, and I believe that they are also very good candidates for immunotherapy. Now, the garden variety endometrioid carcinoma that is not Lynch related, and the papillary serous or high-grade endometrial cancer are also very interesting, but less understood at the moment. That's a very important topic, and so clearly ipilimumab is the front runner of checkpoint blockade therapies, and because of its mechanism of action, which it basically mobilizes the whole immunity across the whole body, it can really trigger, as we have very well established in the clinic, significant autoimmunity. Now, PD-L1 is quite different. Because the target, PD-1, is actually upregulated only in chronically activated T cells that recognize antigen. And so it's more tumor restricted. And so PD-1 blockade or PD-L1 blockade is actually giving us much better safety profiles and enabling now for the first time to really start creating combinations and pushing the envelope. Well, there's no doubt that, sooner or later, a number of these immunotherapy drugs will be approved. And there, I think the important thing is to learn how to use them safely. And second, to learn who to use them for. And so biomarker development will be critically important in this area, much like in other areas of precision oncology today, whereby if we find the right patients, then we can really maximize the benefit while we're trying to minimize toxicity. I think that it's very, very clear that immunotherapy is on the rise, and it will become part of the daily practice of oncology, and therefore it's going to be very important for general oncologists to learn the fundamental principles. But I think the whole field now is mobilizing in terms of educating everyone, and that is a very important and very exciting opportunity. What is very interesting and revolutionary, besides the fact that there is an overall acceptance now that immunotherapy is here to stay in a way, what's also very interesting is that we now have come to realize that most tumors are potentially immunogenic, at least adult tumors, and they could be the target of immunotherapy if we approach it correctly. So no longer melanoma or renal cell cancer are the only tumors that respond to immunotherapy, but now clearly many other solid tumors, lung cancer and so forth, do respond. There are some tumors that respond less, and there the challenge will be to see why. And clearly I think the emerging notion is that there are overlapping complex immunosuppressive pathways that these tumors are using to evade the immune system, and those will be a little harder to crack. But I think that systems biology approaches and a lot of intense study on these tumors will really enable us to understand the mechanisms. So I foresee in the next decade there will be a sort of tsunami of opportunities for cancer immunotherapy and combinations.